Hello again, everyone. Welcome to Chapter 13 lecture on the peripheral nervous system, which includes the cranial nerves, in addition to things like the spinal nerves and nerve tissue outside of the central nervous system. Some basic functions of the peripheral nervous system is that we are connecting the central nervous system to tissues and glands. We have to bring information in, and we also have to send information and commands out. So we have two functional divisions with regard to that basic function. That is the sensory, also known as the afferent. If you are affected, that means you are impacted by something external to you. So this is the afferent division. And we have the motor, the efferent, the effect. You create an effect when you are doing something manipulating outside of yourself. So that's the sensory and the motor. And within the sensory, we have a couple of subdivisions. We have what are called the somatic sensory fibers, and these are general sensations. So these include pain, balance, taste, olfaction, that sort of thing, position in space, etc., etc. We also have what are called visceral sensory fibers. These normally don't reach the level of conscious perception unless they are particularly significant or there are additional sensory inputs above and beyond the basic information about what's going on. So these are called visceral. These are subliminal. Then we have the motor division, which is sending information and commands out to muscles and glands. That can be a sort of a voluntary situation. We call that the somatic nervous system, where you command skeletal muscle by and large. Or we have the autonomous nervous system, or we call it the autonomic. These are usually done absent of voluntary command. The good thing about the peripheral nervous system is that the basic structures don't change all that much. It's more a function of anatomical location. So we have 12 cranial nerve pairs and we have 31 spinal nerve pairs. The main thing I would say is that you need to know the regions. So we have cervical, thoracic, lumbar, sacral, and coccygeal. All right, those are the regions that I would ask you to be able to recognize as potential regions for spinal nerves. Other than that, things are relatively similar, and we'll try to show you some specifics, but not a ton of specifics. So for instance, what is the structure of a nerve? We're going to talk about that over the next few slides. We also will talk about what a plexus is, because that's a site of dysregulation sometimes that leads to sort of a regional issue. And then we have how many plexuses, or you could say plexi, whatever. In any case, this is the set of nerves that innervate the upper and lower extremities and or systems in the chest and abdomen. There is a fair amount of interlacing going on. That is to say, muscles may receive input from more than one spinal nerve and so forth. So there is a little bit of crosstalk in these things, and that's partly what makes it somewhat complicated. The primary anatomical driver of this cross weaving, if you will, are known as as a plexus. That's a structure that allows for crosstalk. We also see this in the autonomic system. We call it the sympathetic trunk, because again, the sympathetic system is more divergent. That is to say, it's going to mobilize more and more varied organ systems in order to meet the need of a sympathetic stimulus. But this plexus is sort of a divergence, if you will, as well. It allows for multiple nerves and a little bit of redundancy in case one or perhaps more is damaged. But if the plexus itself is damaged, or if there's an injury right in that area, it leads to a little bit of an issue. So there is a drawback in a plexus. As the advantages are a little bit more fail safe, but if you are targeted in that one area for injury, then you can lose multiple functions. And so we don't need to know all the ins and outs of the plexuses. You just need to know the names of the four: cervical, brachial, lumbar, and sacral. And those correspond to the areas near where they are located. Given that there are multiple axons and in some cases multiple directions of signaling, it is important that axons be relatively insulated from either from each other, or especially from potentially conflicting signals. For instance, let's say you want to contract your biceps brachii, so you're just flexing your arm, and you want to do it really, really gently. You're only going to fire off a certain number of motor units in order to move an unweighted hand and arm, if you will, as opposed to, say, trying to move a 35-pound dumbbell, which would require most, if not all, of the motor units in that muscle. Well, you don't want to fire off all the motor units accidentally. Okay, that could cause injury if it's an unweighted arm. And you don't want to accidentally send signals down the sensory part of the nerve. So there's a lot of what we would call insulation between the myelinated axon in this case, and either other axons or even other directional neural pathways. All right, so we have this endoneurium, which surrounds an individual axon, and that acts as an insulator from the other axons, again, to allow for some individual function simultaneously, or some function and not others. And you have some fascicle, which is not uncommon. We see that across muscles and that sort of a thing. You've got blood vessels in here because this is living tissue. It needs to be supplied with nutrients, oxygen, remove waste. So we've got the red arteriole and the blue venule, maybe even a little smaller than that, meta-arterial perhaps, I don't know. But these are the blood vessels. And then you have a larger, again, an insulating sheath called an epineurium around the entire nerve. Again, these serve as structural support to prevent damage to the axons if possible, but also electrochemical insulation to allow individual axonal function that is not interfered with by functions in other axons or even other pathways in that same nerve. So we have the structure that we talked about earlier. We have the myelin sheath over the axon, the endoneurium. We've got a fascicle around that, not unlike muscle. This is actually quite similar to muscle, though instead of neurons, what we have would be sarcomeres. Then we have so these are bundled. We have our blood vessels here. Red again is an oxygenated blood. Blue would be deoxygenated. A vein heading back to the heart. And so we have that epineurium around. All right, so we have the endoneurium, the fascicle is the middle one, and the epineurium around the entire nerve. Structurally, we have something here. I probably have mentioned this before, or at least maybe I've mentioned it in another class. I tend to get a little, forget where I said what. In any case, the sensory component of the spinal nerve enters through the back, what we call the dorsal root. And the motor components, the efferent division, come through the ventral root, the root closer to the stomach. And you can tell those two differences, one from the other, by the fact that you have this thing called a dorsal root ganglion. This is where the cell bodies of the sensory neurons are located. So there's a bulge there that you don't see on the ventral root. And so motor comes out the ventral and sensory goes in the dorsal. Again, why would we need so much insulation? Because we have both sensory and motor components in the same nerve. At the downstream end of the nerve would be the tissue, the effector, if you will, the tissue, whether it's muscle or gland, that is going to respond to whatever the nerve is signaling about. In the case of muscle, we have what are called motor units. That is to say, a motor neuron coming down branches out and forms multiple junctions with multiple muscle fibers. This is known as a motor unit. If you try to think of the exercise, it's quite easy for you to sign your name with your dominant hand. And if you practiced a little bit, take a little while, you could get comfortable signing your name with your non-dominant hand. Maybe not as good as the dominant one, but let's get to the real point. If you somehow had a special pencil that you could hook up to the tip of your patellar bone, your knee, all right? So it's pointing out from the knee. There is no way you could ever sign your name the same way that you would sign with your hands. You may be able to do it with your feet. There have been people that have lost arms or have never had arms in the first place learn how to use their feet quite precisely. But if it was your knee, that's anatomically it's just not going to work. It's because the motor units in the knee have a much higher ratio. 1 neuron to 150 muscle fibers, as opposed to nearly 1-to-1 ratio in your fingers or in your lips or your eyes and so forth. So fine motor movement requires low ratio motor units, and gross large muscle groups have large ratio motor units. Let's tie in a little bit of something we've talked about previously when we studied skin in the past. We had different types of receptors in the skin, and most of these receptors are what we call somatic receptors. That is, they are modified nerve endings, modified dendrites. So what happens is they get stimulated. They create a graded potential that then activates action potentials to send to the central nervous system, whether it's touch or whether it's pressure or the nociceptors, which I think are incredibly important because they sense pain and so forth, or thermoreceptors. That's actually a type of mechanoreceptor as well. They send information to the central nervous system, and they are modified dendrites, modified free nerve endings. The special senses, however, use a separate system, and the receptors in those senses are actually dedicated cells. So we'll talk a little more about that. This is an extra slide. This is not in your regular PowerPoint, but I'm including it not as testable material, but to help you better understand the difference between a somatic sensory receptor and a special sensory receptor. Again, somatics are free nerve endings, dendrites, if you will. All of these special sensory receptors, olfactory, vision, hearing and balance, taste, okay, those are mediated by special sensory cells that then interface with neurons. So it's a different system relative to the somatic. Just as a little aside for you to better appreciate the difference. There are a couple of ways we can look at the receptors in terms of classification. It's not meant to make it more complicated, but sometimes you do need to sort of look at them from different perspectives. And the first one would be what we call mode or modality, if you will. That is to say, is it heat? Is it sound? Is it taste? Is it pressure? Is it vision and so forth? And your brain actively has to work on that. We talked about that in the central nervous system. So there are 5, there's some wiggle room here. Some people classify things together, but we'll show you the 5 according to this chart. The first is what we call mechanoreceptors. They can detect physical interaction, if you will, pressure or stretching or that sort of a thing. Now, some people will include thermoreceptors in that because there is a physical interaction there. We will go ahead and break them out as separate. They can detect changes in temperature, cold, warmth, and usually it's relativistic. Now, there are some absolute thermoreceptors in your hypothalamus, but the peripheral receptors are actually what we call relativistic temperature receptors. That's why when you get into a swimming pool that's not quite as warm as you'd like it to be, you kind of creep in and it feels kind of painful almost until you kind of jump in and get your whole body immersed. And then, 30 seconds later, you're just fine. But thermoreceptors detect relative changes, especially when we're talking about the peripheral nerve, the somatic receptors in the skin. Then we have pain receptors, and these are the least differentiated of the receptors because they will activate when a given stimulus becomes too intense, and that means any number of stimuli: heat, cold, pressure, ischemia. There's other things along those lines because they're actually sensitive to chemicals that the cells will secrete when they are undergoing duress. So they're called nociceptors. They're chemoreceptors, but we'll just go ahead and classify them as pain receptors. You have electromagnetic receptors. We call those photoreceptors. I don't know of any other type of electromagnetic receptors. If there is, that would be something I'd learn, but they're photoreceptors. They're sensitive to photons of light emitted by whether it's artificial or natural sources. Then here are the chemoreceptors. These are things like your taste and your smell, or in your hypothalamus, blood oxygenation level, osmolarity, et cetera, et cetera. They can detect the presence or absence of solutes, if you will, in the blood. So that's modality, the different types. You can also classify by structure, whether it's a free nerve ending or it has a special capsule on it. Like, for instance, the Pacinian corpuscle is an encapsulated nerve ending. And then you have specialized sense cells. These are whole cells. Also, location. So you have external ones, you have internal ones, and then you have some that are actually embedded in the tissues. So internal may be around and inside your abdominal or your pleural cavities, around your heart, whereas proprioceptors are for position and location. For instance, just as an experiment here, hold your hand out to your side and wiggle your fingers. You can sense whether your arm is straight or flexed, and you can feel your fingers, and it feels outside of your head. You're not perceiving the sensation inside your head. It feels three-dimensionally outside of you. That's what proprioception is. We've already been over this territory just a little bit. That is to say, we have the afferent division. These are the sensory receptors, and they come in through the dorsal root, as indicated here in blue, all right, with the dorsal root ganglion. And then we have the motor, the somatic motor, which are the voluntary commands that come out through the ventral root, all right, and lead to muscle generally. Then we have a different efferent pathway called the autonomic. I say autonomous. These are the ones that are not necessarily controlled voluntarily. That may include pathways to organs and smooth muscle and very, very rarely voluntary muscle. One big exception I know of with voluntary muscle would be the diaphragm in the separating the pleural and the abdominal cavities. That's skeletal muscle that is controlled involuntarily. So a couple of different efferent divisions: the voluntary and the autonomic, the autonomous nervous system. Only one set of sensory receptors. Although the spinal cord does have somewhat different functional role than, say, the organ system of the brain, Nevertheless, the tissue is still nervous tissue and therefore can be classified in similar ways as to the structures that we saw in the brain. Just for orientation purposes, this is the dorsal side. This is towards the back. This is the ventral side towards the front. Two structures that are quite easy to recall based on the brain structures. One is we have a small little crack here on the dorsal side. This is a sulcus. And on the ventral side, we have a much deeper crack going all the way to near to the center. This is a fissure. We also have this thing called a commissure. It's a little tube going right down the middle. That's a unique structure for the spinal cord. Not unlike the ventricles that we see in the brain, but much smaller. Another thing about this picture that I want you to be careful with, this central tissue region is the gray matter, and it's sort of accurately colored here as gray if you would see an unstained section. But this other material out here that's dark blue has been stained for myelin, and normally myelin is white, so this would be called white matter outside of the gray matter, and that's primarily axons. So let's look at the structures of gray and white matter. We're going to start with the white matter first because it's a little more simple. We have these columns. So here's a dorsal column. It's elongated like a columnar cell. And here's another dorsal column here. There is more finer structure. Some people will get more specific about it, but for our purposes, we have a column here and a column here, the dorsal columns. We also have ventral columns down below as well, and we have lateral columns, right, on the side. So dorsal, ventral, and lateral columns. In the gray matter, we have horns, and we also have dorsal, lateral, and ventral horns. There's a dorsal over here, lateral and ventral horns. So that's pretty much it. All right. So let's ask a couple of questions here. Where does the sensory information enter? The afferent pathway that comes in through the dorsal side. All right. The dorsal root ganglion, easily identified. Dorsal is the sensory side of this equation. What about motor? And this is this is the somatic motor. This is the voluntary stuff by and large. That comes out of the ventral horns. So the ventral horns give rise to the somatic motor and voluntary motor functions that we observe. Then the third question, where do the sympathetic nervous system efferents originate? These are the commands going out to the sympathetic system like heart rate and breathing rate and vascular tone and so forth. Well, those come out of the lateral horns, all right? The lateral horns are where the somatic motor, not somatic motor, the autonomic commanding motor neurons do their thing. So most of us know, or at least have a sense of what it is to have a reflex. We've experienced it in our lives or as patients and so forth. Let's define it now in a standard way. It's generally involuntary. It's often predictable and inborn. That is, a true reflex is a feature of the neuroarchitecture. You stimulate, it gets integrated, and it leads to a dedicated response. It's actually a function of the patterning of the nervous system. That makes it extremely clinically useful for signs of injury or pathology. Now, not all quote-unquote reflexes are inborn. There are ones that are learned, cerebellar type reflexes. A great example of this was a former university president that I worked with talked about going to a haunted house that they were having on campus at the time. And a student jumped out from somewhere in a dark hallway to try to scare the group that he was in. And he said it was all he could do to not turn around and just strike him like a boxer, just literally clock him. And that's a learned reflex. That can be learned through training. It can also be learned through behavioral issues or lifestyle or circumstances and so forth. There are also reflexes that will change with age. Either they go away. Like for instance, the Babinski reflex, where you stimulate the bottom of the foot, it usually goes away by 1 year. The Moro reflex is where if you have a baby and you're holding it and you kind of drop it just an inch or two, just in a jerky like way, the arms will go out. And that reflex was designed to protect the vital organs of the baby, better to injure the upper extremity than to injure the rib cage. And usually that one goes away by 6 months, Babinski by 1 year. I have seen patients, adult patients, where they had a significant cervical injury where the Babinski reflex returned. And so there, when you do neuromuscular assessments, sometimes you're predicting a loss of a reflex, and then sometimes you're looking for the return of a reflex. Most of them are through the spinal cord, cranial nerves, that sort of thing. All right. But they are in this arc structure. That is an afferent to an interneuron, usually more than one. But here we have the simplest of a mammalian reflex, one interneuron leading to a motor neuron that then commands a response. Patellar reflex, et cetera, et cetera. Most mammalian reflexes are generally more than 3 neurons. There are even some reflexes in amphibians and reptiles that are 2 neurons. They don't even have an interneuron here. It is literally the stimulus goes directly to an effector. If you've ever done a dog where you stroke the side of the belly, then they start raising their rear leg and scratching. That's a form of reflex. And so we can classify reflexes as either being what we call somatic, that is activating a skeletal muscle. A tendon reflex is an example of that. Or we have what, shall we say, involuntary reflexes. These are autonomic type reflexes that activate smooth muscle, cardiac muscle, glands, along those lines. So the pupillary response, that's a visceral reflex. There are also subdivisions of the somatic reflexes. They have the stretch reflex. Again, the patellar reflex is a perfect example of this. A flexor reflex or a crossed extensor, that just means that it's that you're pull, like for instance your bicep. You'd pull on your bicep and then relax your tricep. That's usually like, again, a pain or a withdrawal type situation, and we have some examples of that coming up. The third one is what we call a superficial reflex. A little harder to show you exactly. I do have an example of this. It's not unrelated to, for instance, a dog that is kicking the leg when you stroke the belly in a certain way. You can do this in humans as well, and we'll show you some examples of that. So we have here a simple stretch reflex. This is the patellar tendon. They call it a monosynaptic reflex, so there are clearly more than one synapse in here. If you were to look at a frog or an amphibian, you would have a sensory neuron and then a motor neuron, maybe one more beyond that. In any case, this is about as simple as it gets in a mammalian system. So you stretch the tendon either with a knee hammer strike, or I've even seen neurologists use a sort of a karate chop with their hand. That causes the stretch receptors in the muscle to activate action potentials. We send signals up through the dorsal root, where it's integrated not only to the effector of the quads, it will also lead to an inhibitory response in the glutes. So what you get is the result of a kick, because if you didn't concomitantly relax, you would have rigidity rather than a relaxed kick. And here's an example of a flexor, aka withdrawal reflex. If you have a painful stimulus leading up through the dorsal root, interneuron, ventral root to an effector, in this case a flexing action, withdrawing the upper extremity. On the left, you have the test that will show, for instance, in a younger patient up to about a year, you get this toe up response. That's the Babinski sign, and that's also a sign of potential cervical or spinal injury, where you get the reflex that occurs that is normally being suppressed. Under normal circumstances, what you would do is you would test the foot and then get a plantar reflex. You get a downward projection of the toe. That's more normal. Another superficial abdominal reflex, if you will, is where you use either like a tongue depressor or something that's not sharp, and you stroke a side of the umbilicus, and you'll get a contraction of the abdominal muscles. I think being ticklish means that you have higher functioning superficial reflexes than somebody who's not ticklish. One of the areas of some of the most vigorous and aggressive research in neurophysiology is the ability to regenerate following injury. The issue with neurons is that they are so specialized that they can't de-differentiate in order to divide. A cell that is actively dividing by physical circumstance has to be de-differentiated. It has to look more like an egg, a roundish cell that has a nucleus. It has to look more like that than the long projections of dendrites and axon, that sort of a thing. So because they're so differentiated, they just don't divide. So how do we restore if the cell isn't in fact killed outright? Most common site of injury in neurons would be the axons and or the dendrites. But the dendrites are not myelinated. The axons are myelinated. And again, we talk about concussions as a diffuse axonal injury. That's because by and large, because those long axons are a long lever across a fulcrum, they are more prone to focusing energy and creating injury. In the central nervous system, there is some, but it's very limited because what we understand is that the oligodendrocytes actually suppress regeneration of an injured axon. And astrocytes will sort of form a fibrotic, type scar tissue. Now you can actually see these on MRIs and CTs and so MRIs and CT scans. Schwann cells, on the other hand, are really, really interesting because they actually do support regeneration. They can actually restore axonal structure even in partially severed situations. And so there is a growing body of knowledge with regard to axonal injury in peripheral tissues. And again, with the central one, the key would be how to modulate the signaling. I'm sure that if you could, because we know that there are neurons in the central nervous system that can regenerate. So how do we turn those signals on is the extraordinary Nobel Prize winning type mechanism that people are looking for. We're a little more than halfway through this chapter, and so I'm going to say time for a break. So I'm going to terminate the video here because I know it can be hard to kind of slog through hour-long videos or even beyond half an hour or so. So I'm going to take a break right here. This will be the end of part 1, and we will resume in part 2 with the rest of the chapter 13 lecture. Talk to you soon.